Please explain in detail digestive system, such as lipid digestion, carbohydrate digestion, and protein digestion in the format of enzyme, production site, function site, and hydrolysis reaction. The human digestive system plays a crucial role in breaking down large food molecules into smaller, absorbable nutrients. This complex process primarily focuses on three macronutrient categories: lipids, carbohydrates, and proteins. Enzymes, which are biological catalysts, facilitate digestion by accelerating chemical reactions. In this detailed analysis, we discuss the major enzymes involved in lipid, carbohydrate, and protein digestion, their production and function sites, and the hydrolysis reactions they enable. 1. Lipid digestion. Lipid digestion begins in the mouth and continues in the stomach and small intestine. Enzymes break down triglycerides and phospholipids into fatty acids, monoglycerides, and other components for absorption. Enzyme, lingual lipase. Production site, salivary glands, in the mouth. Function site, mouth and stomach. Hydrolysis reaction breaks down triglycerides into diglycerides and fatty acids, primarily acting on short and medium-chain triglycerides. Enzyme, gastric lipase. Production site, chief cells in the stomach lining. Function site, stomach. Hydrolysis reaction breaks down triglycerides into diglycerides and fatty acids, also targeting short and medium-chain fatty acids. Enzyme, pancreatic lipase. Production site, pancreas. Function site, small intestine. Hydrolysis reaction breaks down triglycerides into monoglycerides and free fatty acids, acting on a broader range of triglycerides compared to lingual and gastric lipases. Enzyme, phospholipase A2. Production site, pancreas. Function site, small intestine. Hydrolysis reaction cleaves fatty acids from the SN2 position of phospholipids, generating lysophospholipids and free fatty acids for absorption. 2. Carbohydrate digestion. Carbohydrate digestion starts in the mouth and continues in the small intestine. Enzymes break down complex carbohydrates like starch and disaccharides into monosaccharides, which can be absorbed by the body. Enzyme, salivary amylase. Production site, salivary glands, in the mouth. Function site, mouth. Hydrolysis reaction breaks down starch, a polysaccharide, into maltose, a disaccharide, and smaller oligosaccharides. Enzyme, pancreatic amylase. Production site, pancreas. Function site, small intestine. Hydrolysis reaction further breaks down starch and glycogen into maltose, maltotriose, and alpha-dextrins. Enzyme, maltase. Production site, brush border of the small intestine. Function site, small intestine. Hydrolysis reaction breaks down maltose into two glucose molecules, which are readily absorbed by the body. Enzyme, isomaltase. Production site, brush border of the small intestine. Function site, small intestine. Hydrolysis reaction breaks down isomaltose and alpha-dextrin into two glucose molecules, also suitable for absorption. Enzyme, lactase. Production site, brush border of the small intestine. Function site, small intestine. Hydrolysis reaction breaks down lactose, a disaccharide, into glucose and galactose, both of which can be absorbed. Enzyme, sucrase. Production site, brush border of the small intestine. Function site, small intestine. Hydrolysis reaction breaks down sucrose, a disaccharide, into glucose and fructose, which are absorbable by the body. 3. Protein digestion. Protein digestion begins in the stomach and continues in the small intestine. Enzymes break down proteins into smaller polypeptides and eventually into individual amino acids, which can be absorbed by the body. Enzyme, pepsin. Production site, chief cells in the stomach lining, as pepsinogen, an inactive precursor. Function site, stomach. Hydrolysis reaction cleaves peptide bonds in proteins, breaking them into smaller polypeptides in the acidic environment of the stomach. Enzyme, trypsin. Production site, pancreas, as trypsinogen, an inactive precursor. Function site, small intestine. Hydrolysis reaction cleaves peptide bonds on the carboxyl side of basic amino acids, breaking polypeptides into smaller peptides and aiding in their complete digestion. Enzyme, chymotrypsin. Production site, pancreas, as chymotrypsinogen, an inactive precursor. Function site, small intestine. Hydrolysis reaction. Cleaves peptide bonds on the carboxyl side of aromatic amino acids, breaking polypeptides into smaller peptides for further processing. Enzyme, elastase. Production site, pancreas, as proelastase, an inactive precursor. Function site, small intestine. Hydrolysis reaction. Cleaves peptide bonds on the carboxyl side of small, neutral amino acids, breaking polypeptides into smaller peptides. Enzyme, carboxypeptidase A and B. Production site, pancreas, as procarboxypeptidase A and B, inactive precursors. Function site, small intestine. Hydrolysis reaction. Sequentially removes single amino acids from the carboxyl end of peptides, enabling their absorption as individual amino acids. Enzyme, aminopeptidase. Production site, brush border of the small intestine. Function site, small intestine. Hydrolysis reaction. Sequentially removes single amino acids from the amino end of peptides, further breaking them down for absorption. Enzyme, dipeptidyl peptidase IV. Production site, brush border of the small intestine. Function site, small intestine. Hydrolysis reaction. Cleaves dipeptides into individual amino acids, allowing the body to absorb and utilize them. In summary, the digestive system relies on a complex interplay of enzymes to break down macronutrients, lipids, carbohydrates, and proteins, into simpler, absorbable forms. These enzymes are produced in various locations, such as the salivary glands, stomach, pancreas, and brush border of the small intestine. They function primarily in the mouth, stomach, and small intestine, catalyzing hydrolysis reactions that target specific bonds in macronutrient molecules. This coordinated process allows the body to absorb essential nutrients and utilize them for energy, growth, and maintenance.